Done. And whatever uh, uh, codes that are uh, we discussed how to plot what is done and how can plot about GG plot. So uh, um, we will be discussing the codes for each of these lectures where we will cover like mean and how to discuss the answer. But if most of lying within 50 itself, and maybe 1 or 2 after 50, or maybe no points are there. So you would have this X. So for the function that we use is X limit, uh, or X limit, and then the starting value and the ending value. Okay, so if there, there are no doubts in this, we can move on to the second question. Let's go to the next question then. So in the second question, they have asked to calculate the interquartile range for a series of numbers. Uh, I just saw the chat box about the mic. I've changed the mic now. I hope it's better now. Okay, so let's discuss the second question about how to calculate the interquartile range. So to calculate the interquartile range, first screen is not visible. For me, it is saying that I am sharing the PowerPoint. Let me reshare it. Yeah, I think now you should be able to see it. So we were discussing the answer to the second question about finding the interquartile range for these numbers 10, 20, 40, and so on. So first we have to arrange the data in ascending order from the smallest to the largest. So there are three tens, those come in the beginning. There are three 20s, and then 30, 40, 50. And then we have to find the quartiles. That is, the first quartile would be the 25th percentile, these four numbers. So for these four numbers, we have to find the median, which is 10. So this is Q1 or the first quartile, and then we have to find Q3 or the third quartile, which will be the 75th percentile. So that is the last four values, which will be 20, 30, 40, and 50. So for this, the median would be, because there are two numbers in the middle of the series. To find the median, we have to take the mean of 30 and 40, which would be 35. So we have found the first
Excuse me,
quartile and yes.
ma'am. Actually, the screen is not visible.
Um, okay, I just reshared it.
Okay.
Okay, I'll instead of now I'm sharing the presentation itself on Teams. So hopefully that should be better.
Yeah, ma'am. Now
Okay,
it's
now
fine.
you are able to see it. Okay, okay.
Yeah, yeah.
Thanks.
Thank you.
Yeah, so we were discussing the second question, but I think here a different presentation is being shared. Okay, so to get the interquartile range, first you have to arrange them in ascending order and find the first quartile, which is the mean of the first four numbers. So when you arrange in ascending order, the first four numbers would be 10, 10, 10, and 20. And the median would be 10. So we have the Q1. Similarly, we have to find Q3, that is the median of the last four numbers when they are arranged in ascending order. So that would be the median of 20, 30, 40, and 50. So the median would be um, 30 plus 40 by 2 or 35. So now we have the Q1 value, which is 10, and the Q3 value, which is 35. So now you have to calculate IQR using the formula. So according to the formula, first we have to calculate Q3 minus Q1, which is 10. So I'm just putting the answer in the chat box because somehow here in this a different presentation is shared on Teams, not the one that I was showing. Yeah, so finally, when you apply the formula, the final answer that you will get is 25. So here we have just applied the formula to get the answer to this. So let's go to the next question. So you can take a couple of minutes to read and understand the question and the options, and then you can type the answer in the chat box. Then we'll discuss the solution. The third question is about heat map. The question is which of the following statements is correct for a heat map? So in a heat map, the basic idea is that you show the value in color. So lower values will have a lighter color and higher values will have a darker color. So that is the meaning of a heat map. So the correct answer is option A. A heat map represents numerical values in colors. And option B and C are talking about showing the clusters and a dendrogram for the cluster. So these are additional options in a heat map, like we will see in some of the codes that we discuss. So if we want, we can include the option to show a dendrogram and the clusters, but it is not a compulsory part of heat map. So option B and C are not correct. And option D is also not correct because in a heat map, we will show the entire data in the form of color. So it is not used for dimension reduction. So the correct answer to the third question is option A. A heat map represents numerical values in colors. So let's go to the next question now. Again, you can take a couple of minutes to read and understand the question and type the answer in the chat box. Then we will discuss the solution. So in the fourth question, there is a code given, and there are some options given about the output of the code. So in the code, the function that they have used is called ggplot, and the first argument is data. So it means that the data set which is called as data will be used for plotting the function. And here we are defining what will be the x value of the plot. So in the x-axis, you will have concentration, and in the y-axis, you will have the absorbance. And then the next input is geom underscore point. So we are telling the code that it has to plot it as points. That is, so then it means that it will be a scatter plot where the x-axis is absorbance, y-axis is absorbance. And in the option, they are asking which statement is wrong. So option A, this is a scatter plot. Option A is correct. Option B, the vertical axis is not correct because we have said x is concentration, x-axis or the horizontal axis will be the concentration. Option C, this code uses ggplot2 package, so that is correct. This ggplot function is a part of the ggplot2 package, so option C is correct. Option 4, this code will plot the original data without any statistical transformation. So option D is also correct because if you look at the function, we have directly said x as concentration and y as absorbance. We have not defined any statistical transformation. So whatever input we have given, it will plot it directly. So the correct answer to the question, fourth question is option B. Option B is the incorrect statement. Okay, so I hope this was clear to all. Any doubts? Okay, if there are no doubts, we can move on to the fifth question. So the fifth question is about a volcano plot. So a volcano plot is used to represent changes in gene expression. So in the x-axis, you have the log fold chain, uh, fold change, or how much the gene expression changes, whether it is doubling, whether it is becoming 4 times, etc. And in the y-axis, we have the corresponding statistical significance. So in the y-axis, you have log of the p value. So in the x-axis, the log is to the base of 2. So it will say whether the gene expression has doubled or become 4 times, something like that. But y-axis, the log base is 10. Yeah, I'm sorry. I went to a different question. Um, and the question is, what base is used for log scale for the fold change in expression? So this will be on the x-axis, and the base of the log used is log to the base 2. So this is a theoretical, straightforward question. Let's go to the next one. So let's discuss the sixth question now. So it is they have given the first quartile and the third quartile values. The first quartile is 2 and the third quartile is 8. And they are asking which of the following points will be shown as an outlier. So for a box plot, there is a formula to calculate the range of the box plot. So the values that are above or below, which are falling outside of this range, will be plotted as outliers. So the formula is, um, so the lower range formula is Q1 plus 1.5 times of the interquartile range. The interquartile range. So here they have given the Q1 value, which is 2, and So we have to add that with 1.5 times of interquartile range. They have directly given Q1 and Q3 values. So we just have to subtract 8 and 2. So this would be 6. So this 1.5 into 6 would be 9. So the lower limit is 11. Oh, I'm sorry. So this is not plus sign here because we are calculating the lower limit. So for the lower limit, it is a subtraction. So it will be 2 minus 11, which will be, sorry, 2 minus 9, which will be minus 7. So the formula for the lower limit is Q1 minus 1.5 times interquartile range. So it's not a plus, it's a minus. So this is the lower limit. And the formula for the upper limit is Q3 plus 1.5 times the interquartile range. So here Q3 is 2. So we can, if we just substitute the values, Q3 will be 2 plus, um, this would be 1.5 into 6, the same thing we calculated here. So that is 9. So we are substituting the Q3 value from here, 8. And 1.5 into 6 is 9. So 8 plus 9 will be 17. So the lower limit is minus 7 and the upper limit is 17. So if you look at the options, so the values that are in the box plot will be from minus 7 to 17. If you look at the options, 5 is falling within the range. So 5 will be in the box plot. 10 is also falling within this range of minus 7 to 17. 15 is also falling, and but 20 is greater than 17. So it is outside of the range. So it will be represented as an outlier. And yeah, I'll just go back and discuss question 1 again. There was a problem with the mic. So the first question is about which function will you use to set the range of values on horizontal axis of a scatter plot in R. So scatter plot means you have an x-axis and y-axis, and based on the values of x and y. So if we will at those points, we will be putting a dot. So that is scatter plot. So suppose we are having a scatter plot. Initially, the x-axis is going from 0 to 100. But suppose most of our values are lying within the 0 to 50 range. So then we won't want to plot this range. So 50 to 100 is not needed. So then you have to tell the code that it is the x-axis you only want from 0 to 50. So for that, you have to use the xlim or the xlimit function. So in the bracket of this, you have to write the range that you want. So in this case, we want 0 to 50. So here in the bracket, you have to give the input of 0 and 50. So this is for setting the horizontal axis. You have to use the xlim function. So now this was the first question. I hope after this there was no problem. Yeah, so we discussed the sixth question. Let's go to the next seventh question now. So in the seventh question, they have given an R script, and we regarding that there are some options. So if you look at the first line of the code, it is uh, assigning an to the object P1. We are using the ggplot function. So in a ggplot function, like we discussed already, the first input will be the data or from which data frame you want to plot it. So we are telling the code that we want to plot the data, uh, and in the x-axis we want A, and in the y-axis we want B. So that is what the first line is doing. So we are just telling the code that this is the data that we want to plot, and we want this in the x-axis and what we want in the y-axis. But in this line, we have still not told the code what kind of plot we want. So at the moment, P1 is not having any plot. We have just initialized it with the data and the x and y axis. So in the second line of the code, we are saying what is the type of data that we wanted to plot, because here we are mentioning uh, this keyword geom_line. So this means it will point a line plot. So previously we saw with ggplot, we saw geom_point will plot a scatter plot, and geom_line, this is the other keyword, it will plot a line plot. So the plot will be something like this. In the x-axis, we will have A, and in the y-axis, we will have B, and we will get some kind of a line plot because we have written geom_line. So now let's look at the options. Option A, P2 is a line plot with A and B in the horizontal axis and vertical axis. So option A is correct. Option B, P2 is an empty plot with A and B in the horizontal and vertical axis. So this is not correct because P2 is having a line plot, but P1 would be the empty plot. Option C is also not correct. P2 is a scatter plot. P2 is not a scatter plot, it is a line plot. And option D is also not correct, because it is saying that it will be connected by a dotted line. But this argument that we have given, geom_line, it will be giving a solid line like this. If you want to specify the type of line, then you have to use, so within this geom_line, here you have to mention what is the type of line you want. So there you have to write inside the bracket, if you write line type is equal to dotted, then it would be a dotted line. But if you have not mentioned anything, by default, it would be a solid line. So option A is correct. So let's go to the last MCQ of today. So again, in the eighth question, um, they have given a code for two different plots, plot one and plot two. So in both the cases, we are plotting the same data and the same column of the data as well. And in both the plots, we are plotting a histogram, but the only difference is in the bin width. So in plot one, the bin width is 10, and in plot two, the bin width is 100. So bin width is basically in a histogram. So you will be having on the x-axis different bins, like maybe 0 to so in the plot 2, the bin width is 100, 0 to 100, 100 to 200, 200 to 300, etc. And in the y-axis, you will have how many of the data points are lying between 0 to 100. So suppose maybe uh, this is like 4 values are lying between 0 to 100. But between 100 to 200, slightly lesser, maybe 3 values are lying. And 200 to 300, only 2 values are lying. So this is the meaning of a histogram. For different ranges, how many values are lying. So here the bin width is 100, but in plot 1, the bin width is 10. So if the bin width is 10, then we will have a lot more bins. So for 0 to 300, for a bin width of 100, we are having 1, 2, 3. This is 1, 2, and 3. So we are having 3 bins. But if the bin width is only 10, then we will be having a lot more bins. So in this case, we'll be having 300 by 10, that is 30 bins. So because we have 30 bins, then this histogram will be much wider and bigger. So option A, they are saying histogram in plot 2 is bigger. That is not correct, because as you can see, the number of bins are more in the plot 1. So this first plot will be bigger. And option 2, number of bins in histogram is more in plot 2. So that is also not true, because here there are only 3 bins, but in the first plot there are 10 times of that, 30 bins in this specific example. So option B is also not correct. Option C, number of bins in histogram is more in plot 1. So than plot 2, this is correct. Option D, both are histograms, but in plot 2, the data is smooth. So this is also not correct. Usually with the smaller bin size, then the data will be smoother. So the correct answer to the eighth question is option C. So that's it. We have done with discussing the MCQs for today. Now let's discuss the codes. So I'll share my RStudio screen. So all of you can also open RStudio on your computers, and we can run the code together. If you have any doubts, you can ask, and I'll be explaining what is there in each line of the code. So as you can see from the announcement, the codes are available in your NPTEL course web page itself, in the lectures page. You can also download it from the GitHub file. So I hope all of you have the codes. If any of you don't have the codes, please let me know. I will just copy paste it into the chat box, and from there you can copy paste to your RStudio. So I hope my RStudio screen is visible to all. Can someone please confirm if you can see my RStudio screen? Okay, I think there is an issue with screen sharing today. Mm, just give me a minute. I'll share my entire screen instead of sharing just the window. Now is it visible? Now I'm sharing my entire screen. What about the others? Nobody is able to see it because for me it is showing that my screen is shared. I'll reshare again. So I have tried to reshare it again. Now are you able to see it? Can someone confirm if you can see my screen now? Okay, if you have any problem with seeing the screen, you can stop me at any time. So here, the first week, the first lecture of week 4, we are uh, you doing a scatter plot, and here, so first step is to input the data. Here, the data is called DNAs. It is a default data set in R, so you can read it directly by just writing the file name here as DNAs. And to plot the scatter plot, you can use the plot function and write what will be the x-axis, what will be the y-axis. So before that, if you want to see what is the data set like, you can just type D here. So this is the entire data set. So it has these three columns: run, concentration, and density.
Ma'am,
So yes,
ma'am, the screen is not yet visible.
okay. I'll just try to leave the meeting and rejoin then because
Okay.
I don't know what is the problem. For me, it is saying that the screen is shared.
Okay. Yes, ma'am. Now the
Now it's
screen is
visible.
visible.
Okay, okay, thanks. Yeah, so we in the first week we discussed about how to use R to get a scatter plot. So the first step is to input the data frame. So here we are using this data frame called DNAs, which is a default data set. So you can directly write the name to input. And then, so this is the data set here. So it is having these three columns: run, concentration, and density. Now, suppose you want to plot concentration versus density. So you will use this dollar sign, which will to select which of the column you want. So if you want concentration column, you have to write name of the data frame, dollar sign, and name of the column. So that is the format we have to use. So when you do that, the plot will come in the right hand side bottom of the R screen. So let me run this. So to run the code, I am pressing Command plus Enter or Control plus Enter, or you can also press the run here. So when you run it, you can see this plot with concentration on the x-axis and density on the y-axis. Now, suppose you want to make some changes in the plot. Maybe instead of this D dollar concentration on x-axis, you want the concentration in nanogram per ml to be the title. Then for the x-axis title, you have to use this LAB or X label. Similarly for y-axis, YLAB. And so if I run it, it will be clearer now. So now because you have used this here, the x-axis concentration has changed and you have used this here. So the y-axis concentration has also changed. And we have also changed the color. So now instead of initially it was in black color, now it has become blue color. So here there are, if you see the data for each x-axis, you can see that there are 5 lines. Um, so and but in the initial plot, it was just showing them as dots. But in this plot, instead of dots, we are getting line. So that is because we have changed it here using this PCH. And you can change the size of the lines by using CEX. And if you want to change the size of these x-axis title and y-axis title, then you have to use cex.lab. So now it is 1.5. Now suppose I change it to 3, then let's see what will happen. So now you can see that the font size has increased. So here, yeah, I was also saying that for each x-axis, there are multiple points, and that is because if you see in the data frame, there are multiple runs. That's why there are, for each concentrations, there are different lines. Now, suppose instead of uh, giving them as different line, you want to plot it as a mean. So you want to plot the mean of multiple runs. Then first we are finding the D dot average. So for finding the mean absorbance for each concentration across all the runs, we are using this aggregate function. So once you have found the average, then to the data frame, sorry, to the plot, you have to say that you want to plot it from the average. So you will say that from D dot average, you want to plot the concentration, and similarly from D dot average, you want to plot the density. So when you do that, so initially there were 5 points or 5 lines. Now there is only 1 line. And now you can see that, so if you want to change what is in the x-axis and y-axis, then you can use the x limit and y limit arguments. Because right now the x-axis is only going from 0 to 12. But if you want to increase it to 14, then you can use this function. And y limit is going up to 1.5, but you can see that there is a point after 1.5. So you can make the y-axis go up to 2. So this x limit and x lim and ylim will change the limits of the x-axis. what we discussed in the week 1 about how to plot, do a scatter plot with R. And now, so we can also do it with different kinds of data or different types of formats of data. So here we are going to plot a function from an ELISA data set. So you can see how it is from here, or you can also see it by printing it. So in this data set, you have concentration and you have four absorbance values. So since you have four absorbance values like 1, 2, 3, and 4, like it means that you have taken four different readings for the same concentration, and you just want to find the mean of these four absorbance. Then you have to use 2 to 5 because this is the second column. So second, third, fourth, fifth column. So you are choosing second from the second column to fifth column, and you are finding across each row, you are finding the mean. So you have to use the row means function from second to fifth column. You will be finding the means, and that will be saved to absorbance.mean data frame. And now you want to plot concentration versus mean absorbance. So if you want to check whether the means have been changed, say, you can check it by looking at it. So in this data frame, you have four absorbance values, but in the means data frame, you only have one value and 1, 2, 3, 4, 5, 6, 7, 8 corresponding to these eight concentration values. So suppose you want to plot concentration versus mean absorbance, then concentration you have to take from the first data frame, which is this E data frame, the input data frame, and you want to plot the mean absorbance, so that you can take from absorbance dot mean that we just calculated. And the remaining are and then you can also set other kind of parameters. So you have set the x limit to 0 to 14. So x is going from 0 to 14 and y is going from 0 to 2. And now we have done a scatter plot, but now suppose you want to plot a line plot, then you just have to change one thing that, yeah. So you have to use a new variable called type. So if you want both line, so in this plot, you can see that the dots are there and the line is also there. So if you want both, then here you have to write type equal to B. But if you write type equal to L, so let's change it to L and see what happens. If you type, uh, if you write type equal to L, then only the line will be plotted. So this is about how to plot a scatter plot and a line plot. And now let's try to do it for another data set, which is the facts data set. So we have read the facts file into this variable called D. So let's see what is in this file. So in this file, you have three different conditions: untreated, drug A, and drug B, and you have it readings from different phases of the cell cycle: sub G0, G1, S, and G2M. And now suppose you want to make a bar plot for the only the untreated sample. So the untreated sample is the first row. So you have selected the first row here, and you want to make a bar plot of these values in the second to fifth column. So 2 to 5. So this will create a matrix. So you can, if you want to check what is in this, you can just check here. And now do a bar plot. You can use the bar plot you can use the bar plot function. And for the bar plot function, you have to tell what is the data that you want to use, which is this matrix that we created. And you can also set the Y limit. So here it is the percentage of cells. So since it is a percentage, we are setting the Y axis from 0 to 100. And Y label is the name of the plot, which is percentage of cells. So we have got a simple bar plot. So now suppose you want a bar plot for all of the three samples, because here we have just taken the untreated sample, but now you want to compare the untreated samples and the drug treated samples. Then you have to make a matrix for all the rows. So then the first entry here will be empty and the values are in the second to fifth columns. So we have made a matrix for all of the rows and columns of the data. So now with this new bigger matrix as the input, we are going to plot the bar plot. So since there are three different conditions, you have to tell the code whether you want the bars next to each other. So then if you want them, then you have to set beside equal to true. So because you have set beside equal to true, the three bars of the three different conditions, that is untreated drug A and drug B, are next to each other. And you can also mention about the legend where you want it. So if you want it in the top right, you can write top right, and you can also mention what do you want to be displayed in the legend. So that will come here. And now if you want to change the colors, so first you have to define which are the colors that you want. So suppose light pink, pale, and misty rose. And here using the COL, you have to use the COL argument to tell the code which are the colors that you want to use. Sorry, I didn't run this line. Yeah, we have run this line and now, so now the colors have changed. So in the first week, this is what we discussed. Sorry, first lecture, we discussed about how to get a scatter plot, line plot, and a bar plot. So now let's go to the second lecture of week 4. So in the second lecture of week 4, we discussed how to plot a histogram. So here we are using the example of flow cytometry data. Let me just copy paste the file. There is some problem with reading it. So if you're not able to open a file, you have to give the entire path name. Yeah, so now the file has been read. And now if you want to plot the histogram, you have to use the HIST or HIST function. So before that, let's first see what is in the data frame. So it's a large data frame which is having uh, like many columns corresponding to facts data. And now you want to see what is in the column fl2.h. So you want to have an idea of what kind of values are there in the column. Then you can go for a histogram. So here we are specifying the name of the data frame and which column of the data frame we want. So it is making, in the x-axis, you will have different ranges. So between 0 to 200, about 4,000 or between 3,000 to 4,000 values are there. So that is how you have to interpret a histogram. And now suppose you want to change the number of bins because the x-axis is from 0 to 800. But as you can see, from 800 to 1000, the bin size is very small and we can't see what values are there. So there is a rule called Friedman-Diaconise rule. Based on this, it will calculate what is the optimal number of bins to visualize the data. So you can specify that using the breaks argument. And if you want this Freeman-Dyke-Noyes rule, then you have to use is equal to FD. So now the bins have become much smaller. The bin width has reduced and the number of bins have increased and the data has become more smoother now. And based on this data, we want to also set a line. See from this, where is the mean and where is the median of the data. Then you can use this AB line. So it will plot a straight line at the mean of this column. And you can make another second line with a different color red, which will again, so this AB line will plot a straight line. So at the median of this column. And if I run this line, it will plot it at the mean as well. Now, so for a histogram, there are two ways to show the y-axis. The first way is with a count. So it is saying that suppose 800 values are in the 200 uh, at so from the range of 200 to maybe 205, there are 800 values. So instead of plotting it as how many values, you can also plot it as percentage of values or frequency of values. Then you have to use this setting here. And suppose if you want to plot it, so this is showing it as a density function. And now to convert from density to percentage, you just have to uh, divide it by the sum and multiply by 100. So we have got changed the counts column to percentages by doing this. And now if you plot the histogram, you will get the percentage of values. And we can also give a different color using the COL argument. The first code and this is the other code for the second lecture of week 4. Here we are using this insect sprays dataset. So in the dataset, we have count and spray. So there are 3 different types of spray, and for each spray, we have different count values. And the easiest way to visualize this would be in the form of a box plot, because for each of the sprays, we are having different readings of the count. So to get a representation of all the readings, we can use this kind of a box plot. So for a box plot, you just have to use the box plot function. And here you have to write what will be in the y-axis, this tilde sign, and what you want to be on the x-axis. And finally, you also have to write which is the data frame that you want to use, which will be the D that we have read this file into. And now you can also change the make the plot better. So you can give an X label, you can give a Y label or Y axis title, and you can specify the sizes of the fonts. Sorry. And then here you can see it is only going from 0 to 25, but some of the values are higher. So you can make it to go from 0 to 30. And you can give a different colors for the outlier. So here we have specified red, so it has become red color. So these are some of the variables that you arguments that you can use in a box plot. So this was the second week. We discussed about histograms and box plot. So now let's go to the next third lecture. I hope all of you are also running the codes along with me. So lecture 3, we discussed about how to do a heat map using R. So here we are using this data frame called exp.csv. So if you downloaded the folder, it should be in the same folder where you have the code. So it's not opening, although it is in the same. So usually if it's in the same folder, if you just write the file name, it should open. But in case if that doesn't work, you can copy and paste the path name. So I'm copying and pasting the path name. Now it should work. Yeah, so now we have read the data here. And let's see what is in this data. So you can see that it is a data frame which has different genes and different conditions. So instead of a data frame, it will be easier for a heat map if we make it as a data matrix. So to convert it from a CSV file to a data matrix, you can use the data.matrix function. And the first input will be the name of the data frame. And the second, you can say whether you want the row names or not. So here we want the row names as well to plot the histogram. Now suppose you want it only for the first, now we are going to plot a heat map. So you can use the heat map function, and instead of, you can, as you can see, it's a very big data frame. So let me show it here. So in fact, it is only showing how many, there is a limit for how much R can display. It has omitted these many rows. So there are too many rows in this exp.data. So let's just plot the first 10 rows for a heat map to get an idea of what it will look like. So we are just going to plot the first 10 rows. Sorry. And these two are used to specify about the clustering, so that we will see in the next line. What will happen if we don't give these values? So first, let us just plot a heat map. So you will get something like this. So here the dark colors will mean higher gene expression, and this lighter colors will mean a lower gene expression. Now suppose, so now this is just a heat map. Now if you want to add the dendrograms as well, then you can either, so there are, you can add the row dendrogram or the column dendrogram. So that will be given by these two functions. Now we have omitted both of them. So you will get both row dendrograms as well as column dendrograms. So these um, connecting lines that you see, they are representing how closely the data sets are. And now if you, instead of um, plotting the absolute gene expression values, in some of the cases for each gene, it is better to scale the values based on the values for that particular gene to get better results. So then you can use the scale equal to column to scale the values based on the gene that we have chosen. So now, as you can see, the values are a little bit more uniform because now we have scaled the values as per the uh, gene or as per the column values. So now it will be easier to analyze and make sense of this data. And now, as you can see, the dendrogram are there for both the rows and the columns. But now, if you want the dendrogram just for the columns, sorry, if then uh, you can. So I initially both were NA. Now only col V is NA. So the other dendrogram will be displayed. So Only in the rows we have the dendrogram, because column we don't have the dendrogram. So this is about the heat map. And in the same week, we also discussed how to do a volcano plot. So volcano plot is used for plotting gene expression data. So in the x-axis, you have fold change in gene expression, and in the y-axis, you have the significance level or p-value. So let's discuss with an example. So first, we are going to use ggplot to make this plot. So we are uh, initializing the library. And now let's take the data that we want, which is vdata.csv. So I'll copy as path. Now I'll try to read it. Now it has read properly. So now let's have a look at what is in this data. I'm just checking if you have any questions. Okay, so I think there are no questions. Um, yeah, so we have read the data, and so this is the kind of data that we have. So we have the gene, the name of the gene, or then here the column name is gene underscore symbol, and you have the log fold change, and the so this will go to the x-axis, and the p-value, this will go to the y-axis. So you can see that some of the fold changes are negative, which means that the gene is downregulated. And one will mean that there is no change in gene expression. And more than one, for example, here there is two. So it means that the gene is upregulated. But here, yeah, they have already done the log transformation. So if the log transformation is done, then positive is upregulation and negative is downregulation. So we have read the data, we have seen what is in the data. And here we are just saying that for here we are saying which is the data that we want to use, which is the data that we have read here. Is this? And we are saying what will be in the x and y value. So as we discussed for a volcano plot, x-axis will be log based to fold change, y-axis will be negative log based in p-value. And we want them as different points. So it will be here we are giving geometric underscore point. And now to show the plot, we have to type this. So this is this is the volcano plot, as you can see. Now suppose we want to plot it with a cutoff. Now you can, based on the cutoff, you can draw these different lines. So you can draw the x-intercept and the y-intercept. So the y-intercept we are drawing at p-value of 0.05, which will be the p-value cutoff. So this is the line coming here. So y-intercept will be a line parallel to the x-axis. and we are drawing x intercept line at -1 and 1, or double the times, because it is a log base 2. And now suppose we want to see whether the genes are upregulated or downregulated. So here we are defining a new variable that, or in this data set, we are adding a new column. So let's see how the data is looking after we have added a new column. So initially this column was not there. Now we have added this column. Now we can change the value based on whether it is upregulated or downregulated. So when do we call a gene as upregulated? When the fold change is greater than equal to 1. So fold change 1 means it is expressed 2 times because it is a log base 2. So but sometimes, and the difference has to be significant. So we also have to keep a p-value cutoff of less than 0.05. So if the gene expression changes, uh, log change is more than 1, and if the p-value is less than 0.5, then it will become upregulated gene. But if the fold change is less than -1, or it is the expression is becoming half, and the p-value cutoff is also satisfied less than 0.05, then it will be downregulated. So now if you see the data, yeah. So although there are many genes which are greater than 1 and less than 1, so we can see that still they are coming under no. That is, no means no change in gene expression because the p-value cutoffs are not satisfied. Now, if you take something, maybe this one, which is in up. So here it is in up because the p-value is e power -20. So the p-value is small enough. And the log fold changes 2, which is greater than 1. So it is an upregulated gene. Now, in this volcano plot, we are interested to give different colors to the upregulated and downregulated genes. So here we have to uh, write color equal to differentially this column, because based on what are the values in this TIFF expressed column, we will give different colors to the gene. So the values in these columns are uh, you have to consider it is in alphabetical order. So down means red color, down regulated, and then the next will be no, which is black, and finally up, which will be green color based on the alphabetical values. So now you can see some red points and some green points. Now we also want to know which exactly these points, which genes they correspond to. So you want to add the label as well. So we are first describing a new column corresponding to the gene label. So initially we have just assigned NA, but now based on the values, based on different conditions, we will be assigning the values. So now along with the data points, the gene labels are also uh, coming up in the plot. And now if you see the data frame also, you will be able to see the new column differentially expressed. So whenever there is downregulated genes, only then the gene label column is filled. When there is no statistical significance or no upregulation or downregulation, then it is NA that we have defined here. And in the next line, whenever this column is not equal to no, then the gene symbol column will be assigned to the gene label column. So that is the meaning of this line, because we only want to write the names of the upregulated and downregulated genes. So that is why we have given this condition that it should not be equal to no. And here we have given label equal to this column, gene label column. So only whichever is having the value will be displayed, but the NA values will not be displayed as the label. So that is the idea behind this. So we have covered how to get a volcano plot under heat map. So moving on to the next lecture. And we have many files open, so I'm just clearing it. So here also we are using ggplot and reshape. So here we will see what all different things we can do using the ggplot2 package. And we are using this ELISA data set. So again, the file is not reading properly. So I'll give the entire path name here. Just check if anybody has any questions. I hope there are no questions, and I hope you are also running the code side by side. So if you have any problem with running the code, you can stop and ask me later. So now we have loaded the libraries, the packages, and, and we have got the data frame as well. Let's check the data frame. So this is the same. No, this is the ELISA data frame, which has concentration and what is the value of untreated and the treated sample. So here we are just, so this is the function that we will use ggplot. And the first step is to uh, use the function, and then you have to give the first input to the function, which is the data frame that you will use, so D1. And then you have to tell the function what you want on the x-axis. So on the x-axis, we want the concentration. And what we want on the y-axis, we want the prefix. So even when you do this, you see that the plot is just starting to come, but there are no data points, or there is also no y-axis. We have not defined it. So and here in step 4, we have defined x and y-axis. Still the data points have not become, because we have to tell the code what kind of data we want. So whether we want a point or whether we want a line. So for a point, we have to use geom_point. So now we can see that the points have also come, because we have told the code that we want it as a point. Mm. So now it is the plot is directly displayed here, but you also have the option to save the plot or to assign the plot to a variable. So you can, for example, here we have just done P1. And now if you type P1, the plot will be displayed. So now P1 is already having this plot. And on top of this, if you want to add a line, you can just do P1 plus geom9. So on top of the plot, the scatter plots that we have, we will also get the line plot because we have used the plus here. And now you want to plot both the treated and untreated data. Then you can use the melt function. So let's see what the melt function has done. So if you use the melt function here, then initially the treated and untreated, it has made them both into a same column so that it will be easier to plot. So if you compare this with B1, initially untreated and treated were two different columns, but after melting, untreated and treated are in the same column. Now this second column, instead of calling it as variable, you can call it as sample type because the sample type is treated or untreated or sorry, sample year. So now we are making the same plot with the melted or P2 data frame. So X is the concentration, Y is the absorbance, and the column will be sample. So that means that based on the sample type, it will make two different plots. So this same plot is having two types of data points, treated and untreated with different colors. And this is saved in the P3 plot. So to the P3 plot, if you add a smooth line, you can use geom underscore smooth. So now it has made these lines as well. So you can see that for the treated values, the absorbance is greater than the untreated values. Now you can also edit the x-axis and y-axis. So here to the apart from the P4 plot, we are adding the labels and the colors. So now so here the syntax is a little different. Initially, just for using the scatter plot or line plot, we just used xlab, but here there is a function called labs for the labels, and within that, for x label you have to use x, and for the y-axis label you have to use y. So there is a slight difference in the syntax to use, so please keep a note of that and try to remember it. And now if you want to change the y-axis limit, then you can use the y-line function. And there are also different themes. So if you have a classical theme, then you will get a standard plot with lines for the x-axis and the y-axis. So here we have done it by adding it one by one, but you can also do it all together by first giving the data and then the x-axis, y-axis, and the color values. So you have to color equal to sample, which means it will give different colors for the different samples. And you can set the size and add this smooth line. So you can just do a plus here and give all the inputs. So that will be the final plot. So if you do the final plot, it will be the same here. So this was about using the GG plot. So this is the lecture 6 of week 4 and the last code that we will be discussing today. So in this lecture, we discussed how to use statistics with GGP. So first we are loading the package and we are reading the file. Give the path name here. This file, so the file has been read. So we can check what is in the file there. So it's having many columns with different parameters of facts. So first, let's do a histogram that we saw in the, I think, third lecture. But here, instead of using the histogram with R, we are making the histogram with ggplot. So after giving the input variable and the x-axis, we have to give the plot type, which will be a histogram. And we have assigned it to this variable facts.plot1. So if you type this, then you can see the plot here. And now suppose we want to reduce the bin width, then within the geom underscore histogram, we can use this function, bin width equal to 5. Plotted, so now the bin width has reduced. So if you compare plot 1 and plot 2, in plot 2, the number of bins are more and the bin width is less and the plot is more smooth. Now we can also do other kind of things like giving the label. So in ggplot, we already saw that for labels, you have to use the labs argument with x and y for the x label and y label. And for the y-axis limit, it is the same YLIM, and you can set the theme and text size and which um, font to use. So here we have just plotted the histogram directly without assigning it to the variable. So we are done with plotting the histogram. Now we want to calculate some statistics as well. So let's use this insect sprays data, which we already saw. There are different types of spray, and for each spray, there is a count value. Now we will make a box plot with ggplot. So earlier we saw how to make a box plot directly with R. Now we are seeing how to make box plot with ggplot. So the first um, uh, argument is the data set, which is D that we have right here. And then using the AES variable, you can show which variables you want on the X and Y axis and by which color you want to sort the variables. And finally, for a box plot, you have to use GEOM underscore box plot. So for, if you remember, for a histogram, here we use GEOM underscore histogram and ggplot. Here we have to use GEOM underscore box plot. The others, again, LABS, we have discussed 8 times now. It is for the X label and the Y label. And you can set the Y limit and you can set what is the theme you want and the font size and the font type. So as you can see, the different type of spray are having different colors because here we have given color equal to spray and the outliers are having the red color. Here we have given outlier dot color equal to red. And we can also change the outlier shape. So if I double it, then you will see that the red dot will become much more bigger. And here we have set show dot legend as false. If you do true, then you will also see the legend for which color is for which true. So now you also have a legend here. So this is for a box plot, and in a box plot, we also discussed in the questions directly that it will automatically plot, find the Q1, Q3 interquartile range and median. So this line will usually be at the median, the line inside the box plot. Other option is to also find these values manually. So here for that, let's use this iris example. This was this is a flower data set with information about petal and petal width, etc. This data frame we have already used in the previous classes as well. So now we want to get a bar plot with the mean values of the petal length. So then we can use the ggplot function, and this is the input data set that we have read here, bar.data. And for the different species, so x-axis will be the species, we want what is the petal length. And because we are doing a bar plot, it will not show these values that we see in a box plot. So we have to get it by specifically mentioning it in the code. So to do that, we can use the stat underscore summary. So let's see. So it is looking like this. So, and if you want to see what this is doing, you can just remove this. And as you can see, nothing much has changed. That is because this line is just doing it is getting the summary statistics, but it is not, we are not including it in the plot. So we will here. Since how many packages are installing, so let's wait for that. So that's why the plot was not displayed properly. I have to install things. Yeah, so we have looked the data and we have got the statistics summary, but right now we have just got the bar plot. So to add the summary, like the error bars, you have to use this argument geom equal to error bar, and you can also set its width. Here we just did bar of the means. So we have got the bar plot. But if you do error bar, then instead of this, we will be getting the error bars. So we have a bar plot, and on top of the bar plot, we have the error bars. And now if we want to change the layer arrangement, then the initial parts are the same, what we did already. Um, and you can also set the other X label and Y label, etc. That also we discussed already that we have to use the LABS function for the X and Y axis names and Y limit for initially to 0 to 6, we can change to 0 to 8. So now this will be the final plot with all of the information. Okay, so that's it for today's session. We have revised all the lectures and what was covered, the code that was covered in the lecture, and we discussed some problems. So if you have any doubts, you can ask. Otherwise, we can meet for the next week session. If there are no doubts, we can end the session for today and meet next week. Okay, great. So I'll see you all next week.